Hello, all. Thank you for joining us for this video discussion we're having today, where we'll be talking with Dr. Peter Hersh about CTAK, corneal tissue addition keratoplasty. I'm Bernie Iliakis, President and CEO of CorneaGen, and specifically, we'll be asking Dr. Hersh to give us some background on the development and the history of the procedure. As an introduction, Dr. Hersh is Director of Cornea and Research Division at the Cornea and Laser Institute in New Jersey, which he founded in 1995. He's also a clinical professor of ophthalmology at Rutgers University Medical School. He's a graduate of Princeton University and Johns Hopkins School of Medicine. Dr. Hersh completed his residency and corneal fellowship at Harvard Medical School. And pertinent to our discussion here, he's had a long-time passion and focus on refractive surgery and keratoconus. And as such, he is the originator of CTAK, corneal tissue addition keratoplasty, an amazing treatment for keratoconus. Welcome, Peter. It's great to have you here.
Thanks, Bernie. Thank you for having me.
I have a few questions for you about the history, as we've been obviously had a very successful launch of the product, and sometimes it's good to go back and figure out how do we get here in the first place. And so, first question, CTAK has generated an amazing amount of interest in the cornea community since we launched it just a little less than a year ago. So we're happy to get a chance to discuss CTAK in general with you and specifically hear from you about what drove the genesis of this procedure. So, please share with us.
I have been interested in keratoconus for my entire career. And in the mid-teens, had been working a lot on corneal cross-linking for keratoconus. And cross-linking turned out to be a very, very important procedure for decreasing the progression of keratoconus. But it became quite clear at that time that patients had a real need for improved vision. Yes, cross-linking could stabilize the cornea, but patients were severely debilitated vision-wise. Having worked with preserved corneal tissue in the past, I thought that potentially using corneal tissue inlays could improve the corneal optical surface and enhance a corneal topography. So at that time, we received IRB approval. That was back in 2015 to use preserved corneal tissue inlays to try to improve corneal topography.
Well, that's really interesting. Obviously, it has developed over the next 10 years to be what it is now. So if I get this accurately, you had been ideating on this solution basically since 2015 with a femtosecond prepared inlay. Is that correct?
That's right. So what we did was this. In the office, we would prepare with our own femtosecond laser corneal tissue inlays to specifications that we evolved from research work looking at corneal topography in KC. First patient that I treated was in 2016. We originally started with larger corneal lenticules that we shaped and implanted. What we found early on was that we were getting good corneal topography results, good flattening of the cone, better symmetry of the cornea, but patients weren't seeing that much better. And we attributed that to the tissue interface. So what we started to do was essentially remove the center of the lenticule and make more of a donut-like configuration. This was subsequently evolved again with our team, with Drs. Greenstein and Gellies, into the CTAK procedure that we see today. So it really has undergone almost a decade of evolution.
That's great. I know there has been obviously a little bit of chatter out there of its origination in the 2018, 2019 range, but clearly you have been working on this for a decade. So it's good to know our history. And speaking of that, it's clear there's a lot of excitement around this solution, but there are some other solutions out there that it can be compared to. Can you speak to that a little bit?
Sure. Well, if you look at actual inlays for keratoconus, the first that was used were intracorneal ring segments, Intacs and Kera rings, and they indeed had some success. There are some issues with biocompatibility, and certainly they were not customizable to the individual patient. You may have heard of the CAIRS procedure. That's an acronym for corneal allogeneic intracorneal ring segments. So CAIRS is essentially a tissue mimic of the intracorneal ring segment procedures. CTAK was really developed in an entirely different way because we were looking at doing an intrastromal lamellar keratoplasty procedure to improve corneal topography and vision. And this has, again, evolved to give us extremely good results in our study. Patients improved, on average, 6 lines of their uncorrected vision and indeed have some kind of a wow effect. The next day, they are seeing a lot better. The other important attributes of CTAK are, number one, we are able to use a custom approach using patients' corneal topography and tomography data. Currently, we're using the Pentacam, but along with CorneaGen, we are trying to expand this to other topography and tomography instruments. We're able to take individual corneal data, translate that into an appropriate custom inlay, which is then prepared by CorneaGen using your standard eye bagging techniques, tissue preservation techniques, to yield a preserved, shelf-stable inlay, which can optimize the patient's visual acuity. Important in this is indeed, in addition to the customized approach and the meticulous preparation of the tissue, we're also able to give the surgeon a suggested plan to optimize placement of the CTAK inlay, as well as its location with regard to the cone and with regard to the pupil. We have found long-term that the CorneaGen inlay tissue is biocompatible, very clear early on, and maintains clarity over a long period of time. In addition, the gamma irradiation makes the tissue very easy to use. It has a degree of stiffness to it that lets the surgeon apply it with fair ease. We think of CTAK as really being a surgical ecosystem. We have meticulously prepared tissue. We have specifications defining the custom tissue, specifications defining placement of the tissue, a surgical line of equipment designed to make this procedure something that's of fair ease for the surgeon and the patient. So the surgeon can embrace this ecosystem again, and I think get excellent patient results very quickly.
No, absolutely. And it's certainly that. I think it's that comprehensive nature that have made so much excitement around it from surgeons, and to the point that our wet labs that we have and our trainings are booked, and now 60 plus surgeons that have done at least one, and dozens in the waiting to continue with this great comprehensive solution.
And we're getting excellent feedback from the surgeons. I think they're very happy with the results, and more importantly, the patients really appreciate the results.
Well, thank you so much for your time today, Peter. Really enjoyed the time together and sharing more about the history and evolution of CTAK. For more information and to integrate CTAK into your practice, please contact us at ctac@corneagen.com. You can also check out our extensive library of great educational content, surgical pearl videos, and recorded webinars at corneagen.com. Thank you all very much, and thank you, Peter.
Thank you, Ernie.